We've been talking about hydrochloric acid, and now we know that lots of stuff is going on inside of the stomach. There's all kinds of mechanical digestion, and during the mechanical digestion, the stomach is going to manipulate with hydrochloric acid and other juices and some enzymes and create this cream of mushroom consistency kind of stuff that's called chyme, and that the chyme will be spit out of your stomach just a little bit at a time. We've learned that hydrochloric acid is necessary for breaking up connective tissue, like any kind of ligament or gristle that's in the food that you eat, plus the cell walls of plant, turning the iron that's in your food into something your body can more easily use, killing bacteria, and that hydrochloric acid actually activates a couple of digestive enzymes. One of them is pepsin, the other one is the lingual lipase. Remember, the lingual lipase was in your saliva. It wasn't digesting fat while you were chewing, but when it gets down into your stomach, bumps into the acid, that activates it. Let's talk a little bit more about pepsin, and I want to mention that we are now talking about a group of molecules that are known as Zymogens. I see that is not written here, so let's go ahead and write it down. Let's write down Zymogens. Zymogens are always proteases. Proteases are going to be protein destroying enzymes, right? So Zymogens include pepsinogen. We will be talking in a second about trypsinogen, chymotrypsinogen, and procarboxypeptidase. Good. Okay. All of these are Zymogens. And what's the deal with the Zymogens? Well, first of all, it's kind of nifty that they managed to name the Zymogens in a way that makes it easy for us to remember what's what. The Zymogens are always proteases, and they are made in an inactive form. They are made as a relatively large proenzyme. It's a pretty big protein, and it can't do anything, okay? It's completely harmless. Why? Because most of your body's really important functions are carried out by proteins. So when we have got an enzyme that deliberately destroys protein, it is like having a pet, I don't know, what would be a pet killer robot, all right? You're like pretty sure it's not going to kill you, but hey, why take any chances, right? All of these Zymogens are dangerous proteases, and so they need to be made in an inactive form. And then they get activated by cleavage. And that's where their names came from. So the Zymogens, see, they got enzyme there in the name. The Zymogens, they have got these longer names like pepsinogen, and when they get cleaved, you can think of it as like the O-gen gets cut off of the word, and the enzyme is left behind. This is what happens in the stomach to pepsinogen. I told you that it got activated by hydrochloric acid, and here's what happens. The hydrochloric acid is going to turn the pepsinogen that was made by the glands of your stomach into the active form pepsin, right? Now, what about these other guys? These other guys, trypsinogen, chymotrypsinogen, procarboxypeptidase, they're down in the small intestine. We'll get to them, but they follow the same rules, right? So let's talk a little bit about what cells are making all of these things, okay? What cells are making them? So if I were to show you through the microscope what the lining of the stomach looks like, it kind of looks like this. It's got this simple columnar epithelial tissue, but it is folded up into what gets known as glandular epithelium. And in the glandular epithelium, there's all kinds of cells. I don't really know why they are drawn in these specific ways, but You are not going to be asked to identify them anyway, not by their appearance. You will need to know who does what. Up here near the surface of the stomach, your food would, by the way, would be up here. All your food would be going by. Up here near the top, this is where there are cells that make lots of mucus. That stuff, mucus, is a high protein solution that has got a little bit of stickiness so it doesn't just mix in with the food that you ate. And it has got buffers inside of it that keep this little thin area that's right next to your living cells of the stomach. It keeps this area at a very neutral pH so that pepsin and lingual lipase will get denatured if they dare to get close to your living cells. There are cells called parietal cells, and the parietal cells make the hydrochloric acid. You also need to know that parietal cells make a substance called intrinsic factor. Intrinsic factor, okay? Intrinsic factor is necessary if a human is going to be able to absorb vitamin B12 from their food. Vitamin B12 is required for making red blood cells. No vitamin B12, no red blood cells, right? So vitamin B12 is necessary for that. And yet you could take, like 10 pills of vitamin B12. If you do not have this substance made by your parietal cells, you will still have a vitamin B12 deficiency. Without intrinsic factor, all the B12 you eat just goes right out the other end. And what's interesting is that in some older people, their parietal cells will quit making intrinsic factor. And that is one of the reasons why the elderly can develop a very dangerous vitamin B12 deficiency called pernicious anemia. Pernicious anemia doesn't just cause anemia. That's one of its symptoms. But pernicious anemia also will cause brain damage. So if you have an elderly loved one who has started to become slightly anemic, they need to see their doctor. And generally, this will be treated with a few injections of vitamin B12 to get them back on track, and then monthly injections of vitamin B12. Because if you get an injection of vitamin B12, you don't need the intrinsic factor to help you absorb it. So parietal cells make two things: intrinsic factor, hydrochloric acid. What about chief cells? Chief cells make pepsinogen. They also make gastric lipase, but please remember it's the chief cells that make pepsinogen. Here's how I remember it. I remember, what is the chief enzyme that's working in the stomach? Oh, it's pepsin? The chief cells make the pepsin, okay? So here you go. Sometimes the elderly need injections of vitamin B12. When my parents started getting injections of vitamin B12, I was like, stuff and nonsense. Just eat a healthy diet. And I was wrong, okay? Why does it happen? Because sometimes the elderly, their parietal cells are no longer making intrinsic factor. All right. A summary of the stomach. Good. Right. Let's start talking about more enzymes, okay? We already talked about our first zymogen. I wrote here usually proteases, but they're always proteases. So let me just take that off. They're always proteases. So zymogens, they are proteases, and they are proteases that are being used to digest your food. And since you are basically made most of the active part of you is protein, we can't have these guys loose near any of your living cells. These things are all being made inside of the pancreas. And when they get loose in the pancreas, they will digest your pancreas. It creates a very serious health problem called pancreatitis. So the pancreas, we've already learned about the pancreas. You know now that the pancreas is important for the endocrine system because it makes insulin and glucagon, but it's also important for the digestive system. When it makes enzymes, those enzymes are going to be put into little tubes like this, and those tubes are going to carry it down and into the duodenum. So that function is known as its exocrine function. So the exocrine part of the pancreas is, I don't know, it's like more than 90% of the weight of the pancreas. And these are going to be all of the cells that are busy making enzymes to help you digest your food. So we've got a few more zymogens, which I wrote down on the previous slides. The other enzymes other than pepsinogen are trypsinogen, chymotrypsinogen, and procarboxypeptidase. We're going to talk a little bit more about them. What other enzymes all of these digest proteins. What other enzymes are being made by the pancreas? There's a pancreatic amylase. I asked you to remember that any enzyme called an amylase allows you to digest starch, right? Pancreatic lipase digests triglycerides. We even have enzymes that allow us to dismantle RNA and DNA, and they're ribonuclease and deoxyribonuclease. All of that. Now, let's talk a little bit more about the activation of zymogens. First, we've already learned about the activation of one of the zymogens. What activates pepsinogen? Which one? No, I didn't freeze. Hydrochloric acid. That's what activated pepsinogen. But we don't have any hydrochloric acid in here. The stomach has dumped out its chyme here into the duodenum, and instantly the duodenum has put in bicarbonate, which is going to totally change the pH. So now the pH in here is a pH of about 8 or 9. The rest of the enzymes that work in your digestive tract, they like a pH of 8 or 9. So here's what's going to happen. Trypsinogen is going to come out of the pancreas along with his friends, chymotrypsinogen and procarboxypeptidase. All three of them are out there. Then they're going to bump into an enzyme. See this enzyme in blue? Enterokinase. Enterokinase is known as a brush border enzyme. A brush border enzyme. Why is it called a brush border enzyme? Because the intestines are lined with these little finger-like projections called villi, and on each villus there are even tinier microvilli, and to someone somewhere that looked like a hairbrush. There are enzymes that are actually attached to the hairbrush. I didn't make a point of it, perhaps I should have, but up until this moment, all the enzymes that we've been discussing are made somewhere and then exocytosed from the cells and get thrown into whatever it is that you just ate. Now we've come to enzymes that are called brush border enzymes. Enterokinase is only one of them. And enterokinase is a brush border enzyme who has a couple of jobs, but one of his jobs is to activate trypsinogen. So all three of these guys, trypsinogen, chymotrypsinogen, procarboxypeptidase, they all get squirted out of the pancreas into the duodenum. Trypsinogen is going to get activated by enterokinase and turned into trypsin. And then trypsin will activate his friends, chymotrypsinogen and procarboxypeptidase, and they become chymotrypsin and carboxypeptidase. So we have learned three different methods for activating zymogens. Number one, hydrochloric acid. Number two, enterokinase. Number three, trypsin activates chymotrypsinogen and procarboxypeptidase. And what is going to be the job of all of these zymogens? I think this is a study guide question. It's probably on the exam. Zymogens are proteases, and proteases are going to specifically break peptide bonds. They don't just whack apart proteins willy-nilly, right? Remember, proteins, it's kind of like you put together the pop beads and make it into a little pop bead necklace. Well, those are all individual amino acids that have been put together. We want to take them apart so that they can go into your bloodstream and then they get put back together and made into a different protein. We wouldn't want to take that pop bead necklace and smash it apart with a cleaver. Well, then you can't rebuild it again, right? So all of the proteases, they break peptide bonds, they digest proteins and break them down into the monomers called amino acids. We'll start there at the next video.